Shortly after the Big Bang, which occurred about 14 billion years ago, the only chemical elements present in the universe were hydrogen, helium, and lithium, the three lightest atoms in the periodic table. These elements gradually came together to form stars. These first stars were massive and short-lived, producing all the heaviest elements through stellar nucleosynthesis. Carbon, currently the fourth most abundant chemical element in the universe, was formed mainly in white dwarfs, especially those over 2 solar masses. When these stars reached the end of their life cycle, they ejected these heavier elements, including carbon and oxygen, throughout the universe. These heavier elements allowed the formation of new objects, including rocky planets and other bodies. According to the Nebular Hypothesis, the formation and evolution of the solar system began 4.6 billion years ago with the gravitational collapse of a small part of a giant molecular cloud. Most of the collapsing mass gathered in the center, forming the Sun, while the rest flattened into a protoplanetary disk from which the planets, moons, asteroids, and other small bodies of the solar system were formed. The Hadean Earth was initially inhospitable to any living organism. During its formation, the Earth lost a significant portion of its initial mass, and as a result, lacked the gravity to hold the molecular hydrogen and most of the original inert gases. The atmosphere consisted largely of water vapor, nitrogen, and carbon dioxide, with smaller amounts of carbon monoxide, hydrogen, and sulfur compounds. The oceans may have appeared as early as 200 million years after the formation of the Earth, in a warm environment at nearly 212 degrees Fahrenheit. It is thought that the solution of carbon dioxide and water made the seas slightly acidic, giving them a pH of about 5.5, which then moved towards neutrality in a few million years. 4.3 billion years ago, the Earth's landscape was quite different from today. The Earth was a watery world with little or no continental crust. Its extremely turbulent atmosphere was subject to intense ultraviolet light from the young sun, cosmic radiation, and continuous impacts from asteroids and comets. It was at this time that the resulting internal heating of the core and mantle would have caused the creation of many smaller and more active tectonic plates than exist today. 4 billion years ago, changes in the orbits of the giant planets of the outer solar system bombarded the Earth with asteroids and comets, hailing the Moon and the inner planets. This event, known as the Late Bombardment, would have made modern life impossible. Frequent collisions with objects up to 500 kilometers in diameter sterilized the planet's surface, vaporizing the oceans in just a few months. The hot steam resulting from these impacts formed immense clouds at high altitude, completely covering the planet, making photosynthesis impossible. After this event of extreme violence, the rains would have slowly lowered the height of the clouds, bringing the oceans back to their original depth, only 3,000 years after the late bombardment. These impacts would have brought quantities of organic matter. The periods between such devastating events allow time windows for the possible origin of life. If the deep marine hydrothermal environment was the site of origin of life, then life could have occurred only 300 million years after the creation of the Earth, or 4.2 billion years ago. But if the site of the origin of life was on the surface, this life could have occurred only 3.7 billion years ago. If life had evolved more than 10 meters deep in the oceans of that time, it would have been protected from both late impacts and high levels of UV radiation from the sun. Simulations of geothermally heated oceanic crust produce many organic compounds. In the depths, near the hydrothermal springs, heat escapes between 100 to 150 degrees Celsius, and it is precisely at these temperatures at which some organisms have been found alive. These organisms are placed at the base of phylogenetic tree of life, as close as possible to the last universal common ancestor. The first known life on Earth existed more than 3.5 billion years ago, during the Eoarchean era, when a sufficient crust had been solidified. The first physical evidence found so far are microfossils in northern Quebec. This discovery suggests that life developed very soon after the formation of the oceans. The structure of the microbes was similar to that of bacteria found near hydrothermal vents today, supporting the idea that the origin of life, also called abiogenesis, began near hydrothermal vents. The scenario, called the primordial soup, summarizes the process of the origin of life in 4 steps. First, the Earth had a primitive reducing atmosphere. In fact, the Earth, as it cooled, caused the escape of some very light elements from the original magma to form the atmospheric envelope of the time, composed mainly of hydrogen and helium. Secondly, this atmosphere produced simple organic compounds called monomers under the action of various sources of energy, UV light, heat from the mantle, and even fermentation. Third, these compounds have accumulated over time in a kind of chemical soup, which may have been concentrated in various places, coasts and lagoons, hydrothermal mount, etc. Fourth and last, thereafter, more complex polymers and compounds developed in the mixture, eventually leading to life. The first 3 stages have been the subject of reconstructions and laboratory studies, but the 4th stage of this theory can be described as very simplistic, reminiscent of the famous magic wand of fairy tales. So let's go a little deeper into these mysterious steps that make the Earth a unique place. But before that, a brief reminder of the universal language of nature, which constitutes the living. As you probably know, a cell is an essential biological unit that is found in every living being on Earth, except for viruses, but we'll talk about that later. And in this cell, there is a nucleus, which contains almost all the DNA of the cell. The DNA is a very long molecule, composed of a succession of nucleotides attached to each other by bonds. There are 4 different nucleotides, A, C, G, and T, whose order of linking is very precise and which stuck end to end corresponds to the genetic information. This DNA molecule, structured in a double helix, is the support for the genes, which are nothing more than the succession of nucleotide bases. As the cell does not have an unlimited lifespan, it is necessary to save the genetic information. This DNA must therefore be copied in a very faithful way during the multiplication of the cells, during a process called replication. Replication is therefore the mechanism of production of new DNA molecules. At the cellular level, DNA copying results in the formation of 2 daughter molecules that are identical to each other and to the mother molecule. To do this, the 2 strands of the parent DNA molecule are separated, and each serves as a template for the formation of a new complementary strand. This step is called DNA replication and involves a whole cellular machinery to open the DNA, unwind it, copy it, etc. Now that we have saved our DNA, we must also be able to use its genetic information to code future proteins useful for all living organisms. The genes carried by the DNA will be coded in another form, in messenger RNA, during a process called transcription. The information contained in the genes will be used to make thousands of proteins that are involved in the functioning of the cell. The first step consists of copying the information from the genes in the cell in the form of a molecule very close to DNA, the so-called RNA. During gene transcription, one of the DNA strands is transcribed into an RNA sequence by a complex of a dozen proteins. This copy, called the messenger RNA, is destined for the protein factory and provides the recipe for coding these proteins. The reading of the message carried by the mRNA and its translation into protein form are carried out by the ribosome. Its role? To decode information written with 4 letters, A, C, G, T, from DNA, and then A, C, G, U from RNA, into an alphabet of 20 letters. the 20 amino acids making up proteins. It does this by reading the RNA sequence in groups of 3 bases, for example GGC. This triplet, called a codon, indicates an amino acid in the sequence of the protein being made. The correspondence between codons and amino acids is defined by the genetic code and is expressed through a family of particular RNAs, the transfer RNAs. Each tRNA contains the complementary sequence of a codon and carries at its end the corresponding amino acid. Thus, when the ribosome encounters a new codon, the tRNA attaches to it and provides the ribosome with the next amino acid to be incorporated into the protein being made. A protein always starts in the same way, the amino acid methionine, coded by the initial codon AUG. The other amino acids then follow in the order indicated by the sequence of codons on the messenger RNA. The synthesis is completed when the ribosome encounters a stop codon, which indicates to the ribosome that it has reached the end of the protein sequence. The latter is then released to fulfill its biological function in the cell. The whole molecular ballet must, once again, be very efficient, as a ribosome threads the protein beads at a rate of 15 amino acids per second. The cell is equipped with numerous control systems, both to track the occurrence of mutations that would alter the information stored in the DNA and to regulate gene expression. It also ensures that proteins are produced in the right quantities and at the right time by precisely adjusting the activity of ribosomes. But what are the origins of these complex mechanisms? Several models hypothesize the appearance of a primitive metabolism that would have preceded the emergence of RNA replication. The so-called gene-first models were formulated at the discovery of DNA and its role in heredity. In these models, the appearance of the genome preceded the appearance of metabolism. DNA molecules would have existed alone, self-replicating from molecules present in the environment. The origin of life then raises a logical difficulty, that of the interiority of metabolism or of the genetic code. Indeed, a complex metabolism cannot exist without proteins, and therefore without a genetic code that allows them to be reproduced. But conversely, the genetic code has no meaning without the metabolic environment capable of interpreting it. The logic assumes the existence of a polymer that is both self-replicating and has catalytic functions. This leads to 2 different hypotheses. The first is that the first forms of life on Earth used RNA as the only genetic component capable of encoding the necessary information. The second one is even more restrictive since it considers RNA as the first form of life on Earth, i.e., as the first chemical system at the origin of biological evolution. This hypothesis is called the RNA world hypothesis. There is then a gulf to be crossed between the models describing the increasing complexity of a prebiotic chemistry leading to the formation of several dominant nucleotides, and those modeling the modern primitive DNA cell, which seems to require at least a few hundred genes to produce the constituents of a metabolism capable of replication. Only protein enzymes appear capable of catalytic activity, but these proteins are too complex and varied to be duplicated. The primordial soup is initially formed by relatively simple molecules, but its composition becomes progressively more complex as a result of chemical reactions involving the first-generation molecules. The action of the energy source, ultraviolet radiation or other, on these first-formed molecules creates more complex free radicals, capable of interacting with the molecules of first generations to lead by successive generations to increasingly heavy molecules. These molecules, thus formed, can directly react between them, leading to new synthesis. These chemical systems, by duplicating themselves in the form of replicators, have the important property of not being perfect. The replicas produced are not always strictly identical to the original model, and these differences can influence the ability to replicate. Moreover, the chemical species of this system can have limited lifetimes and/or undergo chemical transformations, called mutations. either by the chemical aggressiveness of the environment where they evolve, by thermal agitation, or by external radiation. Finally, these replicators feed on basic chemical species, which form a limited resource because its renewal is slower compared to the exponential growth of these replicating molecules. The prebiotic soup will therefore progressively be populated by different replicator variants, competing for access to the basic resources and whose objective is to make as many duplications as possible with a limited lifespan. In this environment, a variant becomes the majority when it has characteristics that give it an advantage over other variants. Whether it is longevity, fecundity, or reproduction fidelity, in all cases, selection pressure structurally favors the best performance in one of these three forms of stability. From the moment that chemical loops of this type are set up, the prebiotic soup is thus the site of a kind of survival selection, leading to the eventual favoring of a new variant that exhibits better stability, or that reduces the stability of the other replicant variants with which it competes. But life is not just a chemical reaction like any other. It is also governed by the second principle of thermodynamics. There is an essential difference between living beings and clusters of inanimate carbon atoms. The former tend to capture energy from their environment much better by dissipating that energy as heat. When a cluster of atoms is driven by an external energy source, such as the sun, and surrounded by a heat bath, such as the ocean or atmosphere, it gradually restructures itself to dissipate more and more energy. This could mean that under certain conditions, matter inexorably acquires the physical attribute associated with life. The heart of this phenomenon is based on the second principle of thermodynamics, also known as increasing entropy. Imagine taking a box of puzzle pieces and placing them on a table. In theory, it's possible that the pieces would fall perfectly into place and create a complete puzzle when you took them out of the box. But in reality, this never happens, simply because the chances are very low. Each piece would have to fall into the right place to create a complete puzzle. There's only one possible state where each piece is in order, but there are an almost infinite number of states where the pieces are out of order. Mathematically speaking, it is very unlikely that an ordered outcome will occur at random. Similarly, if you build a sandcastle on the beach and come back a few days later, it won't be there anymore. There's only one combination of sand particles that look like your sandcastle. On the other hand, there's an almost infinite number of combinations that do not. Again, in theory, it's possible for wind and waves to move the sand and create the shape of your sandcastle, but in practice, this never happens. The chances are astronomically higher that the sand will be scattered in a random pile. These simple examples capture the essence of entropy. Entropy is a measure of disorder, and there is always much more disorderly variation than orderly variation. Here's the crucial thing about entropy. It always increases with time. Left to its own devices, life will always become less structured, like sandcastles. But you can fight the pull of entropy. You can solve the puzzle, but you will have to expend energy. But as the universe naturally slides towards disorder, you must expend energy to create stability, structure, and simplicity. So particles that move and interact tend to adopt configurations in which energy is distributed. Eventually, the system reaches a state of maximum entropy called thermodynamic equilibrium, where the energy is uniformly distributed. Although entropy increases over time in a closed system, an open system can keep its entropy low by greatly increasing the entropy of its environment. A plant, for example, absorbs extremely energetic sunlight, uses it to build sugars, and ejects infrared light, a much less concentrated form of energy. Thus, the overall entropy of the universe increases as the plant dissipates the sunlight acquired through photosynthesis, even as the plant itself prevents its decay by maintaining an orderly internal structure. This means that clusters of atoms immersed in a bath at a certain temperature, such as the atmosphere or the ocean, must constantly rearrange themselves to resonate better and better with their external environment. Self-replication, or reproduction in biological terms, the process that drives the evolution of life on Earth, is a mechanism by which a system could dissipate an increasing amount of energy over time. And a good way to dissipate even more heat is to make more and more copies of yourself. The underlying principle of this evolution is adaptation based on dissipation of matter, and this principle applies to inanimate matter. In addition to self-replication, greater structural organization is another way in which highly driven systems dissipate energy. A plant, for example, carries solar energy through itself much better than an unstructured pile of carbon atoms. Thus, under certain conditions, matter can spontaneously organize itself. This could explain the internal order of living things and many inanimate structures. Snowflakes, sand dunes, and whirlpools all have in common that their structure facilitates a dissipative process. This theory provides an alternative explanation to Darwinian theory for each adaptation. Thus, the reason an organism exhibits characteristics X rather than Y is not because X is stronger than Y, but because physical constraints facilitate the evolution of X over Y. Basically, life can only exist by exploiting a thermodynamic disequilibrium, and it is the increase in entropy produced by the dissipation of this disequilibrium that appears to be the driving force of life. Life is based on the chemistry of carbon in the presence of water, and mainly involves 5 families of chemical compounds: lipids, the carbohydrates, nitrogenous bases, monomers, proteins. Any theory of abiogenesis, i.e. the origin of life, must be able to explain the origin and interactions of these 5 classes of molecules. The hypothesis of an RNA world, which is now favored by scientists, is based on the discovery that RNA can both carry genetic information, like DNA, and perform metabolic tasks, like proteins. RNA could thus be the first replicating molecule because of its properties. From the point of view of reproduction, this molecule has 2 essential functions: the storage of information and the catalyst necessary for self-replication. It is therefore theoretically possible, on this model, that RNA alone is sufficient to establish a primitive metabolism. However, an RNA capable of self-replication has yet to be discovered. As we have seen before, gene translation is a largely RNA-governed mechanism, which could have originated in an RNA world. Initially, in the absence of sophisticated proteins, translation could only be imprecise. Therefore, it was only possible to synthesize small proteins or proteins with variable sequences. But conversely, the absence of a large protein allowed the genome to be relatively small. Natural selection can then focus on the performance of gene translation associated with RNA molecules, and as before, induces selection pressure towards more longevity, more reproductive speed, and more precision in replication. Better performance of gene translation means that proteins can evolve towards more complexity and specificity, opening the way to new metabolic mechanisms. The crucial step in synthesizing proteins and allowing the appearance of the first cell seems to be the formation of a primitive ribosome, which was subsequently subjected to natural selection and could have evolved by becoming more complex to increase its performance. The ribosome may have appeared before the first cell, and may have had at the time both the ability to improve protein production and the ability to encode the elements necessary for the duplication of this primitive metabolism, of which it still retains traces in the form of a great density of redundant information. From the moment this generalist pathway of protein synthesis is opened, natural selection no longer focuses solely on the enzymatic capacity of RNA strands, but also on the proteins they're capable of synthesizing. Modern horizontal transfer, which is the process in which an organism integrates genetic material from another organism without being its descendant, is the result of very elaborate mechanisms, implemented in particular by viruses. The first prebiotic transfers may have taken a much more informal form of transfer. The entire metabolism of a prebiotic site can thus be duplicated from one site to another. This mixing between different systems, called sites, allows to diversify the metabolisms of a site and to colonize new prebiotic sites, leading to a competition between variants and to a form of natural selection between the metabolisms of the different sites. The predominance of horizontal transfers implies that evolution at this stage does not function by the appearance of new species whose family tree can be traced, but evolves all the sites concerned by exchanges in an evolutionary process from the same stem ancestor. But this uncontrolled form leaves the prebiotic sites vulnerable to the appearance of predatory or parasitic molecules, which exhaust the metabolism of the site and are likely to spread by the same means. Metabolism must reach a minimum threshold of complexity, the so-called Darwinian threshold, for an entity to be able to reproduce itself solely by virtue of its metabolism, and thus pass the critical stage at which vertical gene transfer, or when the organism receives genetic material from its ancestor, becomes the dominant mechanism. Although RNA is therefore at the origin of DNA and the cellular metabolism, the reaction allowing to pass from one to the other is very difficult to realize because this reaction is very expensive in energy. But RNA, being a fragile molecule, it seems unlikely that it can live without the intervention of proteins. Thus, the origin of DNA is likely to be found after the appearance of proteins, which are essential at each stage of its synthesis from RNA precursors within the cell. DNA has a number of advantages over RNA in terms of preserving the integrity of genetic information. First of all, it breaks less easily, because DNA contains one less oxygen atom than RNA. Oxygen can easily interact with the bonds between nucleotides, posing a problem of stability. But the stability advantages of DNA may not be enough to explain its adoption. It would require an additional selective advantage, which may be due to conflicts between viruses and living cells. In this model, the first DNA organism would be a virus. The DNA would give the virus the power to resist enzymes that degrade RNA genomes, a likely defense weapon for cells. The same principle can be found in current viruses, which alter their DNA to resist enzymes produced by the bacteria they infect. Currently, we can observe that the enzymes necessary for the retrotranscription of RNA towards DNA are very present in retroviruses, whose genome is carried by RNA. This hypothesis is also corroborated by the discovery of DNA viruses, which do not contain T nucleotide groups, but U groups. From an evolutionary point of view, RNA would have appeared first, then DNA with the U nucleotide, or uDNA, then DNA with the T nucleotide, or tDNA, which would have gradually imposed itself. The RNA viruses would be here as relics of the RNA world. The uDNA viruses would then have imposed themselves, and finally come the tDNA viruses. DNA viruses could therefore be older than the first DNA cell. The first DNA cell would have borrowed it from one or more viruses under the pressure of an arms race. The nature of the genome of the oldest common ancestor of all living beings, which scientists call LUCA, remains unknown. Was it still part of the RNA world, or did it already have a DNA genome? In any case, LUCA is the result of a long evolution. The genome of the first cells which preceded LUCA was probably made up of RNA molecules and not DNA. The appearance of RNA was itself a late event. Indeed, this molecule does not seem to be synthesized by the simple methods of prebiotic chemistry. RNA would have been preceded by molecules whose exact nature we will probably never know. What leads scientists to think that RNA was before DNA is the result of this observation. The three great lineages of life share only the protein biosynthesis system, while they differ on the DNA replication system. These elements suggests that DNA was invented before Luca, but was not replicated at that time. The mechanisms of replication were invented only later. There are then several hypotheses for the passage of DNA, of viral origin, into living cells. Either this passage occurred only once, or it could have taken place several times, independently in the different lineages. In the first case, the first genes of the replicating enzymes would then have been replaced by those of another virus, thus forming the three lineages. The most probable hypothesis to date is that the three lineages of life find their sources in the replacement of the RNA genome by the DNA genome of three different viruses. This passage from RNA to DNA would be due to the fact that DNA genomes are more stable and therefore less sensitive to mutations. After more than 1 billion years of evolution, this life has put in place one of the most remarkable mechanisms. The first to use it were cyanobacteria, or their ancient name, blue-green algae, which evolved at least 2.4 billion years ago, paving the way for a remarkable transformation. They became the Earth's first photosynthesizers, making food from water and solar energy and releasing oxygen in the process. This oxygen production by cyanobacteria living in the Earth's oceans was probably at the origin of the Great Oxidation, the sudden enrichment of the atmosphere in oxygen 2 billion years ago, making the environment less hospitable for other oxygen-intolerant molecules. After stabilizing at the lower oxygen levels we know today, the population of cyanobacteria declined dramatically. These dead cyanobacteria drifted into the water, causing their bodies to decompose, which probably reduced oxygen levels. So at that time, the ocean was still not a suitable environment for most life forms that need sufficient oxygen. But as the system of life became more complex, other innovations occurred. Although many bacteria could process many molecules, this life did not have the specialized cells needed for more complex bodies. Animal bodies have various cells, skin, blood, bone, that contain organelles, each doing a distinct job. The life present at that time were just single cells without organelles or nuclei to pack their DNA. Something revolutionary then occurred when microbes began to live inside other microbes, functioning as organelles for them. Mitochondria, the organelles that convert food into energy, evolved from these mutually beneficial relationships. It was at this time, for the first time, that DNA was packaged into a nucleus. Cells also began to live together, probably because certain advantages could be obtained. Groups of cells were able to feed each other more efficiently and also to protect themselves better by being larger. Living collectively, the cells began to provide for the needs of the group, with each cell doing a specific job. Some cells were responsible for making junctions to hold the group together, while other cells were made digestive enzymes that could break down food. These clusters of specialized, cooperating cells eventually became the first animals, which DNA evidence suggests evolved about 800 million years ago. Sponges are among these early animals. Oxygen levels in the ocean at the time were still low compared to today, but sponges are able to tolerate low oxygen conditions. Although, like other animals, they need oxygen to metabolize, they don't need much because they're not very active. They feed by extracting food particles from the water that is pumped through their bodies by specialized cells. Because of their hard skeletons, sponges became the first reef structures on Earth. About 580 million years ago, the beginning of a new period called the Ediacaran began, with a proliferation of other organisms in addition to sponges. These very deep-sea creatures with frond-like, ribbon-like, and even quilt-like bodies lived alongside sponges for 80 million years. Their fossil evidence can be found in sedimentary rocks around the world. At the end of the Ediacaran, oxygen levels increased, approaching levels sufficient to sustain oxygen-based life. This increase is probably due to the multiplication of those first sponges that produced oxygen by eating bacteria. About 540 million years ago, most of these Ediacaran creatures disappeared, signaling a major environmental change. The evolution of animal forms, the food chain, and environmental engineering all played their part. Burrows found in the fossil record from this period reveal that worm-like animals had begun to burrow into the ocean floor. These early environmental engineers disturbed and perhaps aerated the sediments, disrupting conditions for other Ediacaran animals. As environmental conditions worsened for some animals, they improved for others, potentially catalyzing a species change. The period that followed witnessed an explosion of new life forms. Along with the new ways of life of the new animals, hard body parts like shells and spines appeared. Hard body parts allowed animals to more radically change their environment. A change also occurred with more active animals with heads and tails defined for directional movement to hunt prey. The feeding of animals like trilobites may have further disrupted the seafloor on which the gentle Ediacaran creatures lived. Unique feeding styles divided the environment, giving way to a greater diversification of life. Many of these strange organisms were evolutionary experiments, like the five-eyed Opabinia, but some groups, such as the trilobites, thrived and dominated the Earth for hundreds of millions of years. Despite all the changes that followed, almost all the existing types of animals were established 500 million years ago, forming the basis of the ecosystems known on Earth today. Another hypothesis is that life first formed outside the Earth. Organic compounds are relatively common in space, especially in remote areas of the solar system where evaporation of volatile compounds is very low. Some comets have outer layers of dark material, thought to be a kind of bitumen formed by a combination of simple carbon compounds exposed to ultraviolet light. The rain of cometary material on the primitive Earth could have brought quantities of complex organic molecules, which would have favored the appearance of life on Earth. But here again, it would be rather a contribution of organic materials more or less complex than the contribution of life itself. A broader hypothesis is the panspermia, a theory according to which the origin of life on Earth would be due to extraterrestrial contamination. Transfers of microscopic living organisms through space to Earth, natural or deliberate, thanks to comets or meteorites, for example. These theories of an extraterrestrial origin do not directly explain how life appeared, because they only postpone the problem. One could also study the question of the origin of life from a different paradigm or point of view. Rather, it is a matter of addressing the question of how and when evolution began, instead of trying to define life and draw a line as to its origin. Even today, no universal definition of life exists. To give you an idea, no less than 123 definitions of life are already proposed. The boundary between living and non-living systems, as well as the moment from which the non-living would have become living, are quite arbitrary. But for some years now, the problem of life is no longer one of searching for the principles that underline it, but has become a problem of a historical nature. The question is no longer what characterizes organisms in relation to inanimate objects, but rather, how did these characteristics progressively become established in the systems we call organisms? Finally, the question would become, how did Darwinian evolution emerge on Earth about 4 billion years ago in a world that did not yet contain it?